Hello everyone. In this video I'm going to talk through how to calculate magnification in biology. Now magnification and microscopy is a really key topic in the IGCSE syllabus specifically, but this is a really useful skill to be able to do. And I'm going to use past example questions from exams in this video. So we'll talk about how you use values for magnification, size of image and size of the actual object that you're looking at, the formula that you need to use to be able to calculate such values. And then we'll talk about actual exam questions and how to approach different ones. So first of all, let's just talk about the formula and then we'll link everything to that. So the formula for magnification. So if you're given an image, for example, of a cell in an exam question, often it will say next to it magnified by X number of times, because clearly they wouldn't be able to show that image the real size it actually is. And you can use this formula to calculate magnification. So magnification is described as being calculated as size of image, and that's the size of the sort of picture or the diagram that you see. Divide that by the size of, now I say size of actual, the actual thing you're looking at. So image over actual, the actual size of the thing that you're trying to find out. And you can use different letters to represent these three key components of this formula. So magnification is the size of image over the size of the actual thing you're looking at. Magnification we're going to refer to as M. Size of image we're going to refer to as I. And the actual size is A. So clearly this formula now becomes M equals I over A. So take the size of the image, divide it by the size it actually is, and that will give you the level of magnification. Now there is a triangle that you can put together. You might have seen these triangles before to remember this. I'm personally not a fan of these triangles because I think it's really crucial to be able to understand how to rearrange very simple equations. And I do have a video called, this where explains how to rearrange simple equations. And I think that's really useful to watch. If there's 4 different variables in an equation, you would need to know how to play around with it to get which value you're looking for. But here's a nice little kind of nifty trick with this one. You may be familiar with this. I am, this little triangle, and whichever one you're trying to work out, you cover up with your thumb. So for example, if I want magnification, if I were to cover up M, that would leave me with I, and then this line represents divided by A. And you can see, for example, if we were to reverse it, if you wanted something else, if I wanted I and cover that up, you can see that is A times M. So let's rearrange this equation and show the different ways you can have it. So M, magnification, is size of image divided by the size of actual. If I wanted I, so just make a little note here, I, that would be the same as saying M times A. And if I wanted the actual size of something, I would do the size of the image divided by the magnification. So that's how you can rearrange this equation, and you can use this little triangle as a nifty tip. If I just were to give a particular example, just a random example, let's say for example we have a spider here, not too good, and let's say that with the real size of the spider. Let's say there's the actual spider, and I'm now going to draw this spider supersize, so a lot bigger. Just a really easy example to begin with. Now, this spider, the diameter, and when you're measuring length, you always go for the widest part unless the question tells you otherwise. Let's say, for the sake of argument, that this spider is 1 centimetre, so 10 millimetres. In biology, we don't really use centimetres. We only really use metres, micrometres, nanometres, etc. And I'm going to talk about how to convert between the two in just a moment. So let's say this spider is 10 millimetres long, and let's say this particular spider across, just for the sake of argument, pick a number, is 50 millimetres long, and you're told to work out magnification. So in this example, magnification, always show your working. That's really crucial as well, that you show how you work things out. So magnification is image divided by actual. Our image is this one here, just the drawing that we have, and that is 50 millimetres divided by our actual, and the actual image that we're referring to, or the actual object we're looking at is this spider here. We said that was its real life size, and the size there is 10 millimetres. Don't always have to show the units necessarily in this part of the calculation, but we do it at the end. Let's just shrink the screen a little bit here just so we can get a bit more information on. So 50 divided by 10 is 5. So our magnification is 5. Now there are no units for magnification, that's really key. But what we could put is a little x for times 5 magnification. And that would make sense. If you look at this here, you can see that is 5 times bigger. The real spider is 10 millimetres. The drawing of it is 50 millimetres. You can just guess that is 5 times bigger. But through this equation, we are showing it specifically, doing image divided by actual. Just a word about converting between units. This only really works, and you get the correct answer, if your units are consistent. So, for example, if I just circle here, we can see that both of these lengths are in millimetres. Sometimes the question will require you to convert between different units. So here's a helpful reminder. Let's shrink the screen, just give ourselves a little bit more room. And in another video on calibrating a microscope, I do refer to this particular method, but let's start with metres. We don't really deal with centimetres in biology a lot. Then we've got millimetres, an even smaller unit, the micrometres, and even smaller than that, the nanometres. Now if I want to convert between one to the other, I'm always having to work by a factor to 1,000. Each of these are either 1,000 times bigger or smaller than one another. And this is how I remember to do any conversion. It's a really easy way to remember to convert, particularly useful in these style questions where exams often require you to convert. Now you can see I've got divided by 1,000 and times by 1,000 on top and bottom. Now this is all you have to remember. If I'm given a value in millimetres, for example, let's take millimetres here, and I want to convert that to micrometres, a smaller unit, I can times that by 1,000. Equally, if I want to go to reverse that, if I want to go from micrometres to millimetres, I divide by 1,000. So if we take our spider, our large spider, let's take this large spider here, and I expressed its diameter as 50 millimetres. That is the same as, so 50 millimetres is the same as, for example, 50,000 micrometres. It seems a lot larger, but micrometres is a much smaller unit. So 50,000 micrometres is tiny, but it's the same as 50 millimetres. It's by a factor of 1,000. So when you are doing these magnification questions, please just be aware of the units that you are given in the images or diagrams that you're provided. So magnification, size of image over size of actual thing that you're talking about, but just be aware of the units and make sure you give the correct units in the answer if the answer wants you to find I or A. If it's magnification, it wants you to find, remember there are no units, you just have to give the number, the actual number, this tiny number of times it's been magnified. So let's put this into a context, let's talk about some real exam questions. So here we've got 3 real IGCSE pass questions. Let's start at the top with number 1. Very basic, you're given a table with different structures in. You can see onion cell, egg cell, palisade from a leaf, ciliated red blood cell. We're given the image size. Notice we're given the size in millimetres again, and the actual size is in millimetres, so we don't have to do any converting here. And we've got over to the right hand side magnification. So you can see sometimes here, and here we're going to need to calculate magnification, but for others we're going to need other values. So let's just work our way through this one. So we want actual size for the onion cell. We're told the magnification and we're told the image size. Now, some particular questions, like the 2 that I'm going to come on to, you will have to take out a ruler and physically measure certain things. But not in this case. You're simply told you want A. So if we just do an example formula for this one, we want A, which is actual. If you remember from our little triangle or from our formula, that was image over magnification. Image is 50, magnification is 1000. So our answer would be, if we do that sum, 0.05 millimetres. Now that's really important. So over here I would write, if I were doing this separately, 0.05 millimetres. And that's really crucial that we write that in. But as I'm just filling in the table, I just need to write the value 0.05. Notice as well that I'm not writing 0.05 millimetres in the box. I don't need to say this because I already have the units here at the top, in the top column. So I just write the answer 0.05. If I follow this down for the next one, for the egg, I need the magnification. That's image over actual. So I'd be doing image 20 divided by the actual 0.1, and I get an answer of 200. We'll just finish this for completeness sake, and then if you pause the video here, you can try these sums yourself and then press play on the video and see if you got them correct. For the palisade cell, we get a value of 250. For the ciliated cell, this value comes out as 0.025. And finally, we get a magnification of 2000. So hopefully you got those correct if you tried this question yourself. Let's talk about number 2. So number 2 says the diagram shows an Escherichia coli, so an E. coli bacterium at a magnification of 20,000. So straight away, circle this. We're told the magnification, so we have M. We have our M value. We've got the picture here of a bacterium. It says, what is the actual length of the bacterium from A to B in micrometres? So let's highlight the unit to make sure we're not getting confused there. Show your working, really crucial. So it's asking you to show working, so you won't get full marks without showing working. So start with your basic equation. Magnification is image over actual. It wants you to find out the actual length. So you need to rearrange that equation to get actual. So actual is image divided by magnification. So the actual is, now this is where you may need a ruler, because you need to work out the size of the image. So physically, the size of the image they're giving you. So you would take a ruler and place it between A and B, and just measure how long that is. Now, I can't give a value right now for this, because on the screen, if I were to take a ruler, it would be very different to if you screenshotted this and printed it, for example. So I can only really show you the principle in this video of what you would do. So whatever value that comes out as, we're just going to call it X for the sake of argument here. It's more about the method and how to approach these questions and the actual value you get for this one. So image over magnification is the size of the actual, and that's what I want. The image is X, whatever you measure that to be, and even if you get a ruler, you could even do this yourself now, from A to B. So whatever that value is, divide that by m, which you're told is 20,000, and that would give you your actual value. And remember, it wants it in micrometres. So that's how you approach this question. M equals I over A. Rearrange because it tells you it wants the actual length. So A equals I over M. You're given M already at the top, 20,000. And for I, the size of the image, you just need to measure that with a ruler. So that seems fairly straightforward. Let's talk about question, so that was number 2. Let's talk about question 3, which is slightly different. So 3 at the bottom says, the drawing has been made from a photograph. It shows an organelle, so it shows a mitochondria. It's a classic mitochondria diagram. The scale bar on this drawing represents a length of 1 micrometre. Calculate the magnification of the drawing, show your working. Now here's my biggest tip. If they give you any question where they give you this particular bar, where they give you a scale bar, you don't need to measure anything on the physical picture itself. They are telling you that this length, so this bar, so in between these 2 brackets that I've just drawn, that actual length on the diagram, if you were to measure it with the ruler, is 1 micrometre in real length. So they've actually given you everything that you need to carry out this equation. So again, let's write this all down. So we want M, so we're going to answer this over here. So we want M, again, image over actual. We want magnification, so we're still looking for M. It wants I, the size of the image. So for this one, you again need to get a ruler, but all you have to do is lay the ruler here where I've drawn this ruler image and just measure the size of that bar. The size of that bar will give you your value for I, which we'll just call X again, and your actual size, well, it tells you that that bar has an actual size in real life of 1 micrometre. So if magnification is image divided by actual, our image is however long you measure that bar to be, our actual is 1, so our magnification will come out as whatever value you choose it, whatever value that comes out as, so that will give you our magnification, and remember there are no units to this. So again, quite straightforward, but you just have to be aware that if they give you this bar that I've put in blue brackets, they are telling you that that length of the bar is actually the value that it has labelled. So all you have to do is take a ruler and measure the size of that bar, nothing on the actual image. You don't have to measure anything on this actual image here. Just measure the size of the bar, divide it by whatever the value they give you for that bar, and that is the image divided by actual, and that will give you a magnification. And you will hit 2 marks because not only will you have calculated this correctly, but you will have shown the formula. So we have a little bit of information about how to calculate magnification and some real exam questions to help put it into practice. Okay, hope all that helps.